Agaricus introduction and habitat. Agaricus is an edible fungus and is commonly known as mushroom. In old literature it is known by the generic name Sauliata. It is a saprophytic fungus found growing on soil humus, decaying litter on forest floors, in the fields and lawns, wood logs and manure piles. It grows best in moist and shady places and is commonly seen during rainy season. It is cosmopolitan in distribution. A. Bisporous white mushroom are common edible mushrooms and are widely cultivated for food purposes. Some species of Agaricus are poisonous, e.g., A. xanthoderma, and some species may cause gastrointestinal disturbances, e.g., A. placomyces, A. sylvaticus. Structure of Agaricus. It can be studied in two parts: vegetative mycelium, living inside the soil, fruiting body or basidiocarp present above the soil and edible in young stage. A. Vegetative structure. Vegetative mycelium is of three types. Primary mycelium. It originates by the germination of uninucleate basidiospores carrying either plus or strain. The cells are uninucleate, i.e., monokaryotic. It is short-lived and becomes binucleate by fusing of two compatible hyphae. Secondary mycelium. It originates from primary mycelium. After fusion of the hyphae of two opposite strains, the nucleus from one hypha migrates to the other and later gives rise to the binucleate secondary mycelium, i.e., dikaryotic. It is long-lived and abundant. Tertiary mycelium. The secondary mycelium A. Monokaryotic hypha grows extensively under the soil and becomes organized B. Dikaryotic hypha into special tissue to form the fruiting body or basidiocarp. B. Fruiting body or basidicarp. The fruiting body appears like umbrella above ground. It is made up of dikaryotic hyphae. These hyphae are called tertiary mycelium. The mycelium is subterranean. The hyphae are septate and branched. The cells communicate with one another by means of a central pore in the septum. It is a typical dollopore septum. Fairy rings of Agaricus. The mycelium of the Agaricus is subterranean. It has a tendency to grow in all directions from a central point to form a large invisible circular colony. The mycelium also increases in diameter year after year, and the being at all times on the outer edge, because the central mycelium dies away with age. When the mycelium becomes mature at tips, sporophores are produced. These sporophores appear in a circle. These circles of mushrooms are commonly called fairy rings because of an old superstition that the mushroom growing in a ring indicates the path of dancing fairies. Reproduction in Agaricus. It may be vegetative, asexual or sexual. Vegetative reproduction. It reproduces vegetatively by its parennating mycelium. Asexual reproduction. It takes place by the formation of chlamydospores or oidea. Chlamydospores are produced which are lateral or intercalary in position. On germination, it gives rise to hyphae. Ooidea may also be formed under certain conditions which are also known to have sexual function in the diplodization. Sexual reproduction. The sexual reproduction is mainly somatogamous or pseudogamous. The sex organs are completely absent and their function has been taken over by the somatic hyphae which are heterothoric. However, a few species of Agaricus, like A. campstris and A. bisporus, are homothoric. Steps involved in sexual reproduction. Plasmogamy. It is the first step in the sexual reproduction of Agaricus. The vegetative hyphae with uninucleate haploid cells from mycelia of opposite strains, heterothoric, or from the same mycelium, homothoric, come into contact and fuse. Each of such fusion results into a binucleate dikaryotic cell. The dikaryotic cell, by successive divisions, gives rise to the binucleate or dikaryotic mycelium. This dikaryotic mycelium is perennial and produces the characteristic fruiting body of the mushroom year after year. Karyogamy. This is the second step in sexual reproduction. This step is considerably delayed and takes place in the young basidium. In it the fusion of the two nuclei of dikaryon takes place. Meiosis. It is the third and last step in sexual reproduction. It takes place in basidium prior to basidiospores formation. Karyogamy is immediately followed by meiosis. Thus, the basidiospores, formed after meiosis, are haploid. Structure and anatomy of basidiocarp. The mature fruiting body can be differentiated into three parts, i.e., stipe, pilus and annulus. I. Stipe. It is the basal part of the basidiocarp. In this region the hyphae run longitudinally parallel to each other. A transverse section of stipe shows that it is made up of two kinds of tissue. Compactly arranged hyparchae in the peripheral region known as cortex, loosely arranged hyphae, with interspaces in the central region known as medulla. Pilus. The stipe at its top supports a broad umbrella-shaped cap called pilus. The mature pilus is 5 to 12.5 centimeters in diameter. From the underside of the pilus hang approximately 300 to 600 strips or plates of tissues known as gills or lamellae. The gills are white or pinkish in young condition and turns brown or purplish-black at maturity. I.e. annulus. It is ring-like structure present around the stipes. Development of basidium. The basidia are spore-producing bodies. The young basidia arise from the terminal, binucleate cells of the subhymeneum layer. As the basidium grows, the two nuclei of the dichorion fuse to form the synchorion choriogamy. The diploid nucleus soon undergoes meiosis to form 4 haploid nuclei. Simultaneously, 4 narrow tube-like structures develop at the top of the basidium. These are called sterigmata, sterigma. The sterigmata swell at their tips and each forms a small, single basidiospore by budding. Thus, 4 haploid basidiospores are formed in a basidium. Out of the 4 basidiospores, 2 are of plus strain and 2 are of strain. The young basidiospore is unpigmented but it develops brown or black pigments at maturity. Germination of basidiospores. After falling on the suitable substratum, basidiospores germinate to produce primary, monokaryotic mycelium which is either of plus or strain. The mycelia of 2 different strains fuse to form a secondary or dikaryotic mycelium. The secondary mycelium develops the basidiocarps.